Well, hi everyone. We're back again to talk about human kidney physiology and to focus on acid-base balance and to ask the question of ourselves, how does the kidney help to support acid-base balance, and what are the fundamental functions of the kidney nephron that allow us to maintain a normal pH under normal circumstances? We saw actually when we talked about potassium how things start to get a little bit complicated, and we'll begin to see again how the maintenance of acid-base balance also requires different functions of the kidney. Passive K secretion was dominantly a distal process forced upon the kidney by functions proximal to it. And now we're going to see how the proximal tubule and the cortical collecting duct are partnering to help maintain acid-base balance. So what we're going to talk about today, I'm going to make some general comments about acid-base balance. We'll talk about buffering of hydrogen ion. And then we're going to focus on two things that the kidney does that play a key role in maintaining acid-base balance by responding to the consequences of an increased hydrogen ion production as a consequence of our moment-to-moment metabolism. The kidney plays a critical role in reabsorbing bicarbonate, and it actually and surprisingly plays a key role in bicarbonate regeneration. So let's move on to think about that. So here again is our sort of our anchor. This is our body fluid compartments turned on its side. We're talking about intake of food that leads to the generation of acid. And typically in you and I, the metabolism generates about 70 millimoles of hydrogen ion daily. And that hydrogen ion depletes buffer bases like bicarbonate. We'll talk about that. And the kidney has a key role in managing bicarbonate in the proximal nephron, has a key role eventually in excreting the hydrogen ion in the distal convoluted tubule. So the kidney filters about 150 liters a day. And the bicarbonate concentration in you and I on average is about 25 millimoles per liter. So if you do the math, 150 times 25, you end up with 3,750 millimoles of bicarbonate is filtered every day. And that's a buffer base, and we have to reabsorb it. So we need to focus up here on bicarbonate reabsorption, and we need to focus on hydrogen ion secretion and the regeneration of bicarbonate that buffers this daily load of hydrogen ion. So here's the case of the day. This is a dialysis patient with diarrhea. This is one of my nightmares that I see with some unfortunate regularity at the hospital in the patients with end-stage kidney failure on dialysis that I look after. 45-year-old man has end-stage kidney disease. That means he's had a disease process that has damaged his kidney to the extent that he cannot maintain his health with a low level of kidney function as a consequence of kidney disease, and he requires dialysis, and at 45 years of age would undoubtedly be listed for transplant. He developed a stomach flu that was characterized dominantly by diarrhea. He's felt so unwell, he went back to bed in the morning, he missed his dialysis. Monday morning, he was in a coma, brought into the emergency room when his family tried to revive him, and he was found to have the following parameters. The first is his pH, and we'll talk briefly about that. His pH was 6.9. The normal pH in you and I is 7.4. So 6.9 is profound acidemia, a profound rise in the hydrogen ion concentration. The PCO2, partial pressure of oxygen in his blood, was 40. So 40 is normal, so we'll see how that impacted on his management, but we'll see how that high acidemia drives ventilation, and so this is inappropriate. And his bicarbonate level is low, and we'll talk about why his bicarbonate level is low. If I saw this patient in emergency, we would be calling this a mixed acid-base disturbance that is associated with profound metabolic acidosis combined with respiratory acidosis. Now, general comments about acid base in the kidney. Some things that you are probably so simple that we really don't have to think about them, but it just sort of sets the stage for us to for us to better understand some of the functions of the kidney in terms of maintaining acid-base balance or homeostasis. So of course, acid is a substance that can donate a hydrogen ion, and a base is a substance that can bind a hydrogen ion. And the concentrations of hydrogen ion vary over a large range, and so as a consequence, we use the pH to describe the hydrogen ion concentration, which is a log scale. pH is the negative log of the hydrogen ion concentration in moles per liter. In you and I, with a pH of 7.4, kind of conveniently, the hydrogen ion concentration is about 40 nanomoles per liter. So what's the pH in different body fluids? So here's the pH, hydrogen ion concentration in moles per liter, blood, stomach, bile, and urine. So blood pH 7.4, representing a concentration of 4 times 10 to the minus 8th mole per liter. The stomach, of course, is an acidic environment, stomach acid, and so the pH in stomach fluid is 1.5. Bile is alkaline, the pH is 8, and the urine actually the kidney has an ability to excrete either an alkaline or an acidic urine. By and large, the kidney's responsibility is to excrete an acid urine. And it's very rare under normal circumstances to be faced with an alkaline urine. The kidney's job is to excrete an hydrogen ion and as a consequence to lower the urine pH. And you can see that this pH here is almost 1,000-fold than the blood. So the question is, where does hydrogen ion come from normally? So one way, and we'll look in the upper bar, is that fatty acids and amino acids come from your diet, shown on the left-hand side here. On the right-hand side, metabolism generates carbon dioxide as well as water, and it also generates keto acids, including lactic acid. And the diet and metabolism together contribute to adding hydrogen ion to, in fact, our total body water, and we maintain a plasma pH in the extracellular fluid of between 7.38 and 7.2. So hydrogen ion comes from our diet. It comes from sugar or carbohydrate, proteins, and proteins containing sulfur and sulfur-containing amino acids. So cysteine and methionine, for example, when they're metabolized to sulfuric acid, that generates a hydrogen ion, 2 hydrogen ions per mole of 2 moles of hydrogen ion for each mole of hydrochloric acid, sulfuric acid. And in the usual North American diet, we generate about 70 millimils of hydrogen ion a day. In clinical terms, we typically think of it as about 1 millimole of hydrogen ion per kilogram body weight. So fatty acids, carbohydrates not so, proteins produce hydrogen ion. And typically the major proteins responsible for that contain the sulfur-containing amino acids. So 70 millimoles of hydrogen ion, if it was added to the total body water daily, would cause a tremendous change in the hydrogen ion concentration of our total body water. 1 to 2 millimoles per liter. So if we take 70 millimoles and put them into roughly 42 millimoles, then the change in 42 liters, then the change in hydrogen concentration will be about 1 to 2 millimoles per liter. Consequence, 10 to the third mole per liter, and the pH would be 3. And that's incompatible with life. So you and I, when we generate those 70 millimoles of hydrogen per day from our diet and from metabolism, have to buffer that hydrogen ion. We cannot allow the hydrogen ion concentration to rise to those astronomical levels because that would influence protein function. So what do? Well, we buffer it, and that's shown with the red arrow. Here there's bicarbonate in the extracellular fluid. It's our dominant buffer. And then we have proteins and phosphates within the interiors of cells which combine hydrogen. And in urine, we use phosphates and ammonia. And we're going to talk a lot particularly about ammonia, when we look at the kidney's ability to bind hydrogen ion and secrete the hydrogen ion. So let's talk about our buffer systems. And what do we mean by a buffer system? Well, a hydrogen ion plus a buffer, of course, in equilibrium prevents the hydrogen ion concentration from rising because this equilibrium shifts this way, which is uncharged. So buffers limit the change in hydrogen ion concentration when they're added to a solution. We've all been in high school chemistry where we've titrated acid-base, measured the pH, and titrated hydrogen ion concentration as we add a buffer. And examples in humans are bicarbonate, monohydrogen phosphate, which is something the kidney uses. We're not going to talk too much about it, and ammonia, which we're going to talk a lot about. And these are the two key ways that are regulated and influence the kidney's ability to maintain acid-base balance. So let's first look at the bicarbonate buffer system. So in this particular case, if you and I are eating and generating hydrogen ion, we have bicarbonate sitting at a concentration of 25 millimoles per liter, then this will combine to form carbonic acid, H2CO3, which will then be converted to water and carbon dioxide. So increasing hydrogen production will decrease the concentration of H2CO3 as we shift this equilibrium to the right, and it will produce carbon dioxide that we can then blow off. And the pH, shown here in the Henderson-Hasselbalch equation, is a function of the bicarbonate concentration over the partial pressure of PCO2. So as bicarbonate levels fall and PCO2 levels rise, you can see that actually this log value will decrease and the pH will decrease, reflecting the rise in the hydrogen ion concentration. So why is the buffer-based system so efficient? Well, because both sides of the equation can be independently regulated. CO2 can be increased or decreased by breathing, and bicarbonate can be increased or decreased by the kidney. If a disorder causes a fall in bicarbonate, for example, in my dialysis patient who developed diarrhea, which is a loss of a bicarbonate-rich solution, and the consequence is acidosis, the lungs can breathe to lower the CO2 and serve to help bring the pH towards normal. So in the presence of a high acid load, we can regulate the PCO2 from our lungs. So high hydrogen output can influence the pH. We have buffers. The ventilation can serve to blow off the CO2, and the kidney can serve to make bicarbonate and cause hydrogen ion output to serve hydrogen acid-base balance. So if indeed the plasma hydrogen ion increases, there are carotid and aortic body chemoreceptors, and there are that can sense that go to the respiratory system, drive the muscles of ventilation, and lower PCO2. And that's important. The low PCO2 will then serve to reduce the hydrogen ion concentration and tend to buffer the consequences. So if you and I have a hydrogen ion load, we'll have a brainstem response driving ventilation, which lowers our PCO2. And with the Henderson-Hasselbalch equation, we'll tend to normalize that bicarbonate partial pressure of CO2 ratio. So then the question is, okay, hydrogen ion acidemia drives ventilation, but what does hydrogen do to the kidney and how do we influence that? So here's the challenge. Hydrogen from metabolism is buffered by bicarbonate. The bicarbonate turns into CO2 and water, and we breathe out the CO2. If we were to do this day in and day out, and there was no mechanism to restore bicarbonate to normal, then there would be a progressive acidemia as our bicarbonate fell each day. So that is the challenge the kidney has. The kidney has to do two things. It has to prevent loss of bicarbonate in the urine, and it has to regenerate bicarbonate that is titrated by the hydrogen ion load each day. Those are the two functions that the kidney plays in acid-base balance. So let's take a look at what the kidney does. So the first thing is that the kidney has to reabsorb bicarbonate. Remember that we've said that if you think about it, a lot of bicarbonate, 3,750 millimoles, is filtered every day. Now, if we didn't reabsorb that, then we'd rapidly deplete our entire buffer base. And that means that any hydrogen ion added to our body fluid compartments would cause a much more dramatic increase in hydrogen ion concentration, or said alternatively, a much greater fall in the pH. So the first thing the kidney has to do is reabsorb the filtered bicarbonate. Remember, bicarbonate is a small solute, and when water moves in response to Starling forces from the capillary lumen into Bowman space, it drags along the bicarbonate, just as it does the potassium and the sodium. So if you and I, once again, are sitting in the glomerular capillary looking at bicarbonate around us, the concentration is 25 millimoles per liter. If you and I could magically move into Bowman space and look at the concentration of bicarbonate in the water that moved from the lumen of the capillary into Bowman space as a consequence of those Starling forces, the bicarbonate concentration would be the same. So each day, because of the high glomerular filtration rate, the kidney nephron is presented with a lot of bicarbonate, and we cannot afford to lose it. Now, the second thing the kidney does, and you might not have predicted that on the basis on first principles, is that it makes bicarb. So if you and I are sitting in the beaker, we're shoveling hydrogen ion into the beaker each day, we're reducing our buffer base, the kidney is filtering that buffer base, reabsorbing all of it so we don't lose it, and then we want to add more bicarbonate to the beaker. That's the second thing the kidney does. And so we're going to look, and then the last thing the kidney does is it has to excrete the acid. And it does so dominantly by using ammonium. So, two functions of the kidney: bicarbonate reabsorption and bicarbonate regeneration. That together, the reabsorption preserves the buffer base, and the regeneration restores the buffer base. Two key ways in which you and I maintain a buffer to moderate the changes in pH as you and I generate hydrogen ion each day during our cellular metabolism. So let's look at bicarb reabsorption by the kidney. So, hydrogen ion concentration, I've said, is 25 millimoles per liter. There's 150 liters of filtrate a day, so you and I filter 3,750 millimoles of bicarbonate each day. That's what we present to the nephron. And the proximal tubule plays a key role in the reabsorption. What's fascinating is even though we filter 3,750 millimoles of bicarbonate a day, there's vanishingly little bicarbonate in the urine. So bicarbonate, readily filtered at the glomerulus into Bowman space and then entering the proximal tubule, is completely reabsorbed by the nephron so that there's virtually no bicarbonate in our urine. I said the typical pH in our urine is between 5 and 5.5. So bicarbonate turns out, and this may really be an underestimate, but for practical purposes, bicarbonate is almost entirely reabsorbed in the proximal tubule. And how does it do that? Well, that's the question we're going to ask, and here's we're going to answer it. So here's the filtrate with sodium and bicarbonate entering. The first thing that happens is that there's a sodium-hydrogen exchanger. And remember, that was the sodium-hydrogen exchanger we talked about when we looked at tubular function and sodium reabsorption in the proximal tubule. So sodium entering the lumen is reabsorbed in major part because of the sodium-hydrogen exchanger. Now, then what's the next step? Well, the next step is the bicarbonate combines with the hydrogen ion. So sodium enters the cell, hydrogen ion exits the cell because of the antiporter activity. And the hydrogen ion then combines with the bicarbonate to form water and CO2. And this reaction to form water and CO2 is facilitated by a luminal-bound carbonic anhydrase. That enzyme facilitates movement of that equilibrium from bicarbonate and hydrogen ion to H2CO3 and then to water and CO3. So bicarbonate is then turned into water and CO2. The next step is that the CO2 enters the cell, diffuses across the cell membrane. And the next step is that the CO2 in the cell combines with water and in a reverse manner, facilitated by an isoform of the carbonic anhydrase that's luminal, the intracellular carbonic anhydrase facilitates the movement of the equilibrium in this direction, and as a consequence, bicarbonate is reabsorbed and regenerated, and the hydrogen ion then moves back to be secreted, so that the reabsorption of bicarbonate is dependent upon sodium-hydrogen exchange. It's dependent upon an equilibrium that shifts the bicarbonate and hydrogen ion to H2CO3 to H2O and carbon dioxide by virtue of a carbonic anhydrase. The CO2 then diffuses into the cell where an equilibrium between CO2 and water is facilitated by a second isoform of carbonic anhydrase to form H2CO3, which then breaks down to form hydrogen ion and bicarbonate. And so as a consequence, the hydrogen tends to recycle and the bicarbonate is moved from the lumen into the cell and then actually is transported in the antiluminal, the basolateral membrane, as sodium bicarbonate cotransporter. So a lot of steps. So the kidney, therefore, proximal tubule cell, number of components: a sodium hydrogen exchanger, carbonic anhydrase, an intracellular carbonic anhydrase, and a basolateral sodium bicarbonate cotransporter. The consequence? The sodium bicarbonate that you filtered is reabsorbed and re-enters the circulation. And this process limits increasingly the amount of bicarbonate that moves down the nephron and returns the bicarbonate to the circulation and doesn't waste it. So to summarize, what we see here are a number of events. Here's our lumen, here's our proximal tubule cell, here's our blood compartment. Here's the sodium hydrogen exchanger 3, key protein, integral membrane protein that is an antiporter for reabsorbing sodium and secreting hydrogen ion. The hydrogen ion combines with the bicarbonate to form H2CO3 and CO2, and the carbonic anhydrase isoform 4 facilitates that reaction and that equilibrium. The CO2 enters the cell. Carbonic anhydrase isoform 2 moves the equilibrium in this direction. The bicarbonate is then secreted by a luminal membrane, a sodium bicarbonate cotransporter into the blood compartment. And all along we have our basolateral sodium potassium ATPase. It actually creates an important role because it maintains a low intracellular sodium concentration that drives the sodium reabsorption. So sodium reabsorption by virtue of the sodium hydrogen exchanger leads to the reabsorption of bicarbonate by virtue of two isoforms of carbonic anhydrase, and then eventually a sodium bicarbonate cotransporter. A pretty complicated process, but what that means is the kidney doesn't waste bicarbonate. It tends to utilize the exchange with hydrogen ion to reabsorb the bicarbonate. So key job, the kidney is not supposed to excrete bicarbonate day to day when you and I are in a circumstance in which we're generating hydrogen ion. So let's talk about bicarbonate regeneration. Now, like the concentrating mechanism, bicarbonate regeneration, in other words, replacing the bicarbonate that was buffered by hydrogen ion in our general circulation, and the CO2 didn't enter the proximal tubule, it was just breathed out by virtue of the low pH driving ventilation from our brainstem. Let's look at that bicarbonate generation. So again, bicarbonate has to be reabsorbed. But the other thing that the kidney does, and it's in the proximal tubule, is it regenerates de novo bicarbonate, not that bicarbonate that was just created, just turned into CO2 and water in the luminal. It generates de novo bicarbonate by a process called ammoniogenesis. And then eventually the ammonium is trapped in the lumen of the cortical collecting duct and excreted. So let's take a look at how the kidney makes bicarbonate. And you'll see right away that this is a pretty complex process, and I want to emphasize just a few things. The first thing is that it utilizes a transporter at the basolateral membrane of the proximal tubule cell that takes glutamine and moves it in to the cytoplasm of the proximal tubule. So this is amino acid transporter. There's one at the luminal membrane, and actually there's one at the there's one at the luminal membrane and one at the basolateral membrane. So glutamine enters the cell. This middle portion here is meant to represent mitochondria. And in the mitochondria, glutamine undergoes a number of metabolic steps that lead to it's a deamination, the generation of ammonium at 2 steps, and the production of alpha-ketoglutarate and malate. And then the malate is transported out into the cytoplasm. So a consequence of glutamine metabolism in the mitochondria of the proximal tubule cell leads to the generation of ammonium. Now, that's one thing. So then what happens? Well, then the other thing is that the last step from alpha-ketoglutarate to malate also generates bicarbonate. The bicarbonate exits the mitochondria, and then the sodium bicarbonate cotransporter in the basolateral membrane returns the bicarbonate. Not returns, but transports the bicarbonate to the blood compartment. So the consequence of glutamine metabolism in a proximal tubule cell, a process termed ammoniogenesis, is to generate ammonium and to increase bicarbonate reclamation or regeneration. Now, what we have to do now is get rid of the ammonium. Ammonium hops on our old friend, this sodium hydrogen exchange protein, and it then is transported into the lumen of the membrane. So, if you think about starting in the middle, the consequence of glutamine metabolism and ammoniogenesis is to restore bicarbonate to the blood compartment and ammonium to the luminal compartment. The proximal tubule therefore regenerates bicarbonate, but also moves ammonium into the lumen. Now, ammonium is a weak acid, and so it dominantly exists as sodium, as ammonium, but it is in equilibrium with ammonia and with hydrogen ion. Ammonia can diffuse across cell membranes, like CO2. and blood vessels. Ammonium does not readily cross cell membranes, and ammonium concentration increases. So if you and I are sitting there and we decide to eat a big steak, and we eat all of that protein, and it contains sulfur-containing amino acids, and we metabolize those amino acids to generate sulfuric acid and therefore hydrogen ions, what we find is that the kidney will respond. So ammoniogenesis can be regulated. And the more acid we produce, by metabolism, the lower the pH. That low pH drives ammoniogenesis. So it's a process that can be regulated. And that if you and I increase hydrogen production via our metabolism, we can increase our ammonium production between 5 and 7-fold. So it can be very dramatic. So hydrogen ion in excess drives a process that helps to regenerate bicarbonate and create ammonium that can then allow that hydrogen ion to be excreted. So let's return to ammonium and ask ourselves, well, how do we excrete it? And that turns out to be a pretty complicated process that actually, to my knowledge, is undergoing active investigation and isn't entirely sorted out, but it depends on a family of transporters. So let's think about what happens. And so here's a cartoon. This is the nephron, the proximal tubule. Here's our loop of Henle, distal convoluted tubular collecting duct. What are the processes that allow that ammonium to be excreted? What we want is 100% of the ammonium generated to be excreted in the urine. And it turns out to be a bit of a tricky process. And the ammonium is kind of interesting in the way it hops on to various transporters or takes advantage of the differential permeability of ammonia and ammonium to be able, in terms of their ability to cross cell membranes. So let's look here, proximal tubule. The ammonium enters the lumen of the proximal tubule by virtue of the sodium-hydrogen exchanger. So instead of a hydrogen ion moving in the opposite direction to a sodium, the sodium entry drives the secretion of ammonium. So the ammonium moves from the cytoplasm into the lumen. And then it travels down the nephron. There's not very much affecting it here, but then it comes into the thick ascending limb. And here's something that's kind of interesting. So the ammonium grabs onto the sodium potassium 2-chloride transporter. And it is moved into the cells of the thick ascending limb by riding that sodium potassium 2-chloride transporter. And then it exits those cells into the interstitium here by jumping onto the sodium hydrogen exchanger in that cell in the basolateral membrane. So what we're seeing is ammonia jumping onto a sodium hydrogen exchanger here. We see it jump onto sodium potassium 2-chloride transporter here, and then we see it move into the interstitial space by jumping onto a sodium hydrogen exchange protein again, getting moved out. So ammonium moves into the lumen, into the thick ascending limb, out of the thick ascending limb, and into the interstitium in the medulla, in the deep medulla, and in the deep cortex and medulla of the kidney. Now here's where kind of an interesting and completely unexpected transport phenomenon occurs. The problem we have now is how do we get the ammonium that ends up in the interstitium into the cell, the collecting duct cell, so that it can be subsequently secreted into the urine. We know it happens because 100% of the ammonium produced ends up in the urine. And it turns out it uses a series of proteins that are called RH proteins. These are transporters. These are integral membrane proteins that are permeable to ammonia and ammonium. And so this ammonium that sits in the interstitium here rides these transporters into the cell. So the ammonium ends up in the cell of the cortical collecting duct. By taking a pretty circuitous route, and relying on a number of different transporters. 1 transporter, 2 transporters, 3 transporters, 4. It takes 4 transporters to get that ammonium into the cortical collecting duct cell from the proximal tubule cell. It's kind of a remarkable pathway. Now, in the cell, you now have ammonium, of course, in equilibrium with ammonia and hydrogen ion. The ammonia diffuses freely across the luminal membrane of the collecting duct because it's permeable to that lipid bilayer. And then what the cortical collecting duct, the intercalated cell, does is it pumps hydrogen ion across the luminal membrane, where in the lumen of the cortical collecting duct, the pumped in hydrogen binds to the ammonia and traps the ammonium in the lumen of the nephron. And ammonium is not permeable to the luminal membrane of the cortical collecting duct cell. So again, the ammonium, which is important to excrete because that excretion enabled the retention of a bicarbonate, enters the lumen of the nephron in the proximal tubule because it uses the sodium-hydrogen exchanger. It exits the lumen of the thick ascending limb and enters the thick ascending limb cells because of the sodium-potassium 2-chloride transporter. It exits the thick ascending limb cell into the interstitium of the kidney by taking advantage of another isoform of sodium-hydrogen exchanger. And then it enters the collecting duct cell by virtue of these Rh proteins. And the collecting duct cell allows ammonia to diffuse into the lumen of the collecting duct and then pumps hydrogen ion. The hydrogen ion then binds in the lumen of the collecting duct. And therefore, because the ammonium is the luminal membrane of the collecting duct is impermeable to ammonium, the ammonium exits in the urine. Pretty circuitous way of getting there, isn't it? So let's take one look at a slide we saw before, and we ask, how does the kidney secrete hydrogen ion? And the dominant way that the kidney secretes hydrogen ion is shown here. Remember when we talked about sodium and absorption in the cortical collecting duct and potassium secretion, we focused on the principal cell. And here we're focusing on the intercalated cell of the collecting duct, and it has an ability to excrete hydrogen ion with a hydrogen ATPase. So it has a luminal integral membrane protein that uses the energy derived from the hydrolysis of ATP to pump hydrogen ion from the cell into the lumen. The ammonia diffuses out, and then the hydrogen ion combines with the ammonia to form ammonium, which cannot re-enter because the luminal membrane is permeable to ammonia, but impermeable to ammonium. And the hydrogen ATPase allows you to get that hydrogen into the lumen, and where it can combine to form ammonium, and then the ammonium is washed in the luminal fluid and excreted into the urine. Now, the luminal membrane has some important permeability characteristics. It allows ammonia to diffuse from the cell into the lumen, but it doesn't allow hydrogen ion or ammonium to diffuse back into the collecting duct cell, so that the ammonium is trapped. And the excretion of ammonium and the retention of sodium bicarbonate in the proximal tubule serves to replenish a buffer, the bicarbonate buffer base, which is regularly depleted by the addition of hydrogen ion via metabolism and intake in our bodies. So let's just summarize this relatively complicated process, but important to remember. Bicarbonate is filtered. I made a mistake here. I used the 125 liters. I'm sorry about that. But in excess of 3,000 millimoles of bicarbonate is filtered, and it's all reabsorbed. And that reabsorption is dependent on sodium-hydrogen exchange protein, carbonic anhydrase in and outside of the cell, and a sodium bicarbonate transporter in the basolateral membrane. We don't want to waste our buffer base. We don't want to pee out bicarbonate. We want to retain the bicarbonate that's filtered, and then we have to replenish the bicarbonate that titrates the hydrogen ion that is produced each day by our metabolism, and that's also a function of the proximal tubule. So bicarbonate is reabsorbed in the proximal tubule, critical. New bicarbonate is generated by a process called ammoniogenesis, which is the metabolism of is the metabolism of glutamine to alpha-ketoglutarate and to malate in the mitochondria of the proximal tubule cells. And the ammonia exits at the luminal membrane via sodium-hydrogen exchange, and the bicarbonate exits the cell at the basolateral membrane by virtue of a sodium bicarbonate cotransporter. Critical ability to trap ammonia in the lumen of the nephron is to excrete hydrogen ion. We have ways of moving the ammonium from the proximal tubule lumen into the collecting duct cell, and we then want ammonia to move into the lumen and be trapped there. And that requires hydrogen ion secretion by a hydrogen ATPase in the luminal membrane of the intercalated cell. As a consequence, we maintain normal bicarbonate levels, and we adjust in response to challenges of metabolism or bicarbonate loss by increasing ammoniogenesis. And the most simple way in which we do that is a direct effect of hydrogen ion on ammoniogenesis. It drives ammoniogenesis and helps us preserve acid-base balance. So, again, we come to this notion. Here's our fluid compartments inverted. We have bicarbonate, we have hydrogen ion, we deplete buffer, but we regenerate bicarbonate, reabsorb bicarbonate, and excrete hydrogen ion. So acid-base balance is exquisitely dependent upon proximal tubule cell function in partnership with specific functions of the intercalated cells and the cortical collecting duct. Filter a lot of water, we filter a lot of bicarb, we reabsorb the bicarbonate, we generate 70 millimoles, and the kidney excretes 70 millimoles of hydrogen ion and regenerates bicarbonate and serves to maintain acid-base balance. Now, just a little bit of by summary, more on the kidney. For every hydrogen ion secreted, one bicarbonate is added back. That's because the reaction to produce ammonium leads to the production of bicarbonate, mole for mole. One ammonium in the urine equals one new bicarbonate. Ammonia is an extremely important buffer. There are others in the urine, phosphate, for example, sometimes as much as 30 millimoles a day, but phosphate cannot be adjusted. Our ability to increase or decrease phosphate as an important buffer in the lumen of the kidney is very, very limited. In fact, probably not easily regulated. However, the kidney can increase the availability of ammonia and ammonium 5 to 10-fold, which gives us an opportunity to increase bicarbonate generation should we be challenged by increased generation of hydrogen ion or increased loss somewhere else of bicarbonate. So variations in acid excretion in the kidney are dominantly dependent on changes in ammonia excretion. So let's go back to our patient. So this is a 45-year-old man, end-stage renal failure, has essentially no kidney function. So by no kidney function, of course, he doesn't have to worry about losing bicarbonate in his urine. He doesn't have any filtration, but that's only one of the features that roles that the kidney plays. He may not filter bicarbonate, but he cannot regenerate bicarbonate. That's the critical issue. On the weekend, he got stomach flu and had diarrhea. Now, it's important to know that diarrhea is a bicarbonate-rich solution. So diarrhea leads to the loss of bicarbonate. Now, this gentleman did not generate more hydrogen ion. He lost more bicarbonate. And you can see that the bicarbonate level was profoundly low. Now, the other issue is that, he was not breathing properly. Remember, we said that severe acidemia should drive ventilation, and it didn't drive ventilation, and that made the fall in pH much more dramatic than if he had been able to hyperventilate, increases minute ventilation, increases CO2 loss. That would have tempered the extent to which the pH declined when there was a bicarbonate loss. Diarrhea in combination with end-stage renal failure has led to some of the most profound acidemias that I've had to treat in clinical medicine in our hospitals. So what's the analysis? Well, patient has severe acidemia. He has a metabolic acidosis because the bicarbonate is low. The failure to hyperventilate in response to metabolic acidosis contributed to the degree of acidemia or the extent to which the pH fell. The diagnosis, unable to regenerate bicarbonate, loss from the GI tract, and respiratory muscle fatigue. So you can imagine that as he was developing this progressive acidemia, he was breathing just like running. And when you start a run, you can breathe much more readily than you can at the end as you begin to fatigue. And so the increased ventilatory demands of early acidemia eventually led to weakening of his muscle and his inability to lower the PCO2. So in a gentleman like this, what do we have to do? We have to treat him with dialysis. When we put someone like this on a dialysis machine, we expose their blood compartment across the semipermeable membrane to very high bicarbonate concentrations, and we can rapidly restore normal pH balance to individuals like this. All right. So that's lecture 7 about acid-base balance in the kidney. What we're going to move on to in the final lecture is a case of uremia, and I'll let you read this before our next lecture. We'll talk about some other functions of the kidney. We're not going to be focusing so much on transport anymore, but we'll be talking about some of the endocrine functions of the kidney that play a key role in maintaining our overall health. So thanks very much, and we'll see you at the next lecture. Bye now.